So I'm sure you were a little briefed last time that there are, sorry, okay, there are two important systems, the parasympathetic and sympathetic system. So what exactly is parasympathetic system? We will begin with that today. Parasympathetic system is also known as cholinergic nervous system. Here, it is known as cholinergic because it has acetylcholine as a chief neurotransmitter. Cholinergic fibers mean all the fibers which release acetylcholine at the nerve terminal. NT is the nerve terminal, and all somatic motor neurons where all is cholinergic transmission present. It is present in the somatic nervous system in the skeletal muscles. It is a very widespread neurotransmitter, acetylcholine, the most widespread and very short-lived. All somatic motor neurons supplying to the skeletal muscles, you have acetylcholine. All preganglionic, parasympathetic and sympathetic fibers, including adrenal medulla. See, everywhere in skeletal muscles, acetylcholine is present. Everywhere in preganglionic, parasympathetic or sympathetic fibers, autonomic ganglia, including adrenal medulla. And at all postganglionic parasympathetic fibers. Somewhere in postganglionic sympathetic fibers also, you have release of acetylcholine. It happens in sweat glands. It is also released in some synapses in the central nervous system. So widespread is acetylcholine, skeletal muscle as well as preganglionic, all ganglia, postganglionic parasympathetic, a part of postganglionic sympathetic also, and some synapses in CNS. Hypothalamus is the major controlling center. So I guess I would like to ask you here, I would you can reply in your answers, you see. So would you reply in your question section that am I audible? Is the presentation going fine? You're able to see everything? Whatever it is. Now go uninterrupted in this class on cholinergic drugs. Now, acetylcholine is a major neurotransmitter which is produced in the cholinergic nerve fibers. Chemically, it is acetic acid ester of choline. acetic acid ester of choline. There are two important enzymes which are involved here. Choline acetyl transferase for the synthesis and acetylcholine esterase for the degradation. Very important enzymes, choline esterase, acetyl transferase and acetylcholine esterase. It is an ester. Acetylcholine, acetic acid ester of choline. There are two chief substrates, choline and acetyl coenzyme A. Choline acetyl transferase is the enzyme that catalyzes the final step in the synthesis of acetylcholine. Acetylation of choline with acetyl coenzyme A happens. This pyruvate, in the presence of pyruvate dehydrogenase, releases acetylcholine coenzyme A. See, this is all the recapitulation of your knowledge of biochemistry. Okay, आपने सब ये reactions पढ़े हुए हैं, इसको याद करिए. Very important ones. Pyruvate to acetyl coenzyme A with pyruvate dehydrogenase. Then ATP along with acetate and coenzyme A will form acetyl coenzyme A. Okay, in the presence of acetate thiokinase. Choline is taken up from the extracellular fluid into the exoplasm of the neuron. Uptake of choline is a rate limiting step. Transport of choline from plasma to the neuron is unique to cholinergic neurons and have transport system. Transport system is the high affinity sodium dependent choline transport protein. You require energy for choline to be taken up in the neuron. It's a rate limiting step. Now synthesis of acetylcholine. Vesicular acetylcholine transporter will take up acetylcholine. It is an ATPase dependent program. Along with acetylcholine, you have other transmitters which might be released alongside it with the release of acetylcholine. They are known as co-transmitters. ATP, VIP, some nitric oxide ions like calcium, magnesium, proteoglycan. All these could be released along with the chief or the main neurotransmitter. These may be called as co-transmitters. Vesicles also have VAMPs as synaptobrevins and synaptogramins. So this also I'm sure you would have read in physiology to some extent. Now release of acetylcholine. Inner surface of neuronal membrane contains some snaps like syntaxin and SNAP24, these are proteins. Calcium through the voltage gated channels enters the exoplasm, causes depolarization, fusion of these vesicles to the axonal membrane and exocytosis of acetylcholine into the synaptic cleft. Now this is the diagram of whatever I have been discussing till now. Acetyl coenzyme A, this choline has got an active transport into the axon, which is energy dependent and a rate limiting enzyme. Choline esterase, this enzyme will help in synthesis of, which I just mentioned, choline acetyl transferase, acetylcholine. Acetylcholine will be taken up inside the vesicle. Inside the vesicle, there are present other co-transmitters like ATP and substance P, and along with this, everything will be released from the synaptic vesicle. Once they are released, there is this binding of these transmitters onto these balls of the cell membrane, and then these neurotransmitters are released outside. Now, whole this diagram could be a little confusing right now, but it is given in most of your books and it shows at various places which are the mediators which can be affecting this release or synthesis, of acetylcholine. See, sodium goes in, choline goes in, and acetylcholine is formed. Well, there is something known as hemicholine which will block the entry of choline inside. Then besamicol is one substance which will block the entry of acetylcholine into the synaptic vesicle. Now, onto the cell membrane are present heteroreceptors and acetylcholine autoreceptors. Heteroreceptors could be of any other transmitter. Autoreceptors is acetylcholine's receptors, which we will be talking about. Some muscarinic receptors, M2 receptor might be present here. We will talk about that. Don't want to confuse you right now. Acetylcholine, which is produced in the synaptic cleft. Now, something known as botulinum toxin will decrease the release of acetylcholine at the cleft, while calcium will enhance the release. This is actually required. Now, what happens to acetylcholine when it reaches the synaptic cleft? It's not going to last forever. No mediator does. But acetylcholine has got the shortest half-life. It will be degraded by the enzyme known as acetylcholinesterases. Acetylcholinesterases. This is the postsynaptic cell. This whole thing is a postsynaptic cell. Acetylcholinesterase will break acetylcholine into various components and its action will finish. But before the action finishes, acetylcholine might get attached to some receptors which are present on the postsynaptic cell wall, or it can also be attached to the acetylcholine autoreceptors, or it can be degraded. So this is the fate of acetylcholine. Since this enzyme acts pretty fast, the action of acetylcholine lasts for a very short duration. Now you saw that acetylcholine is degraded by acetylcholinesterases. Now these acetylcholinesterases are present at the cholinergic neurons, cholinergic synapses, neuromuscular junction, red blood cells. Acetylcholinesterase is present at cholinergic neurons, synapses, neuromuscular junction, and red blood cells. Acetylcholine is the best substrate for acetylcholinesterase. Now then you have something known as butyrylcholinesterase, also known as pseudocholinesterase. You saw that the site for the presence of acetylcholinesterase is cholinergic neuron, cholinergic synapses, neuromuscular junctions, red blood cells. While for butyrylcholinesterase or pseudocholinesterase, it is present in plasma, liver, glial cells and other tissues. Now, this is the pseudocholinesterase. So this is not the best enzyme for acetylcholine breakdown. It is best for butyrylcholine breakdown, or maybe succinylcholine or benzoylcholine to a lesser extent, but to the least extent acetylcholine. Acetylcholine and its metabolites. Of course, acetylcholine will be degraded by acetylcholinesterases by hydrolysis mechanism to choline and to acetate. Now coming to the very important part of this discussion is the receptors. I just mentioned presynaptic muscarinic receptors. So just to come back to the type of receptors, I'm sure you know that acetylcholine acts basically on two major types of receptors: nicotinic receptors and muscarinic receptors. Now we will talk about nicotinic receptors first. There are two main types of nicotinic receptors: NM receptor and NN receptor. Nicotinic muscular means at the skeletal muscles. NN is at the ganglia. Nicotinic receptors are, you I'm sure remember from your knowledge of general pharmacology, that there are two types of there are various types of receptors, and nicotinic receptors are ion channels. Muscarinic receptors are G protein coupled receptors. So are pentameric, ionotropic. The receptor proteins themselves form ion channels. Ion channels are ligand gated. The ligand attaches to the channel and the channel opens. The natural target of acetylcholine and various drugs are nicotine. Therefore, they are known as nicotinic receptors. These are located on the neuromuscular junction at all neuromuscular junctions, so NM, at all autonomic ganglion, hence NN, And these ganglia include adrenal medulla and at various places in central nervous system. So nicotinic receptors could be NM at the neuromuscular junction or at the ganglia, adrenal medulla or in CNS. They all have muscular heterogeneity. Now just to show you this diagram, this is the motor neuron axon terminal. Axon, it's a cell membrane of the axon, this whole thing, this is a cell membrane. This is the synaptic vesicle from where the acetylcholine is going to come out. Now I'm going to show you what is this postsynaptic junction. Synapse, presynapse, postsynapse. This is a photo or a diagram of an ion channel, which is in this case a pentameric ion channel, which is a nicotinic receptor. This is the magnified version of nicotinic receptor. Here it is a pentameric structure. You have acetylcholine binding onto it. The moment it binds, the sodium goes in and the potassium comes out. And the muscle fiber subunits are there. And this is the muscle membrane, cytoplasm. So I'm sure you can now understand where acetylcholine is going to be released as usual, but where it binds is different. This is a nicotinic receptor, which I just showed you the diagram of. It could be NM in the NM junction, neuromuscular junction, or NN in the ganglia. Now, what are these different receptors? Where are they present? What are their functions? What type of receptors they are? Let's study that. NM, I have repeated many number of times now, it is a skeletal neuromuscular junction. Postsynaptic, of course, these are all postsynaptic. We just showed you the synapse. NN is also autonomic ganglia. Adrenal medulla, postsynaptic again. While some nicotinic receptors could be both pre and postsynaptic, mainly presynaptic in central nerves. At the NM junctions, acetylcholine, while attaching to this NM receptor, will cause contraction of the skeletal muscle. Whereas acetylcholine, while attaching to the NN receptor in the ganglia, will cause excitatory depolarization, firing of the postganglionic neuron and release of the catecholamines. NN in the ganglia. Catecholamines will be released. In central nervous system, excitatory presynaptic release of dopamine and glutamine. Mechanism of action. Very clearly, these are ligand-gated ion channels. Ligand in this case is acetylcholine. The moment acetylcholine attaches, there is increase in the cationic permeability, sodium, potassium, and hence the action happens. Action at NM junction is the contraction of the skeletal muscle. Action at the NN junction is excitatory depolarization of the ganglia, release of catecholamines, while there is excitatory presynaptic release in the CNS of dopamine and glutamate. I hope I'm clear here. I move on. Now coming to the various agonists and antagonists of the nicotinic receptor. Agonist at NM is acetylcholine. Of course, it has to be acetylcholine at both. Other agonist at NM is succinylcholine, PTMA, and nicotine. Acetylcholine, nicotine are both, agonists at both the receptors, while succinylcholine and PTMA additionally on NM and DMPP on NN. Anadoxin A at CNS, the nicotinic receptors. Then the, antagonist at NM, it will be D-tubocurarine, and antagonist at the NN, that is at the ganglia, will be hexamethonium or trimethophan. Mecamphetamine is an antagonist at the CNS. Not really much in need, but mostly you should try and remember acetylcholine nicotine agonist at both, along with the along with PTMA and DMPP, respectively, are agonists at NN, NM, and NN junctions. Digibacurarin antagonist at NM and hexamethonium antagonist at NN. Muscarinic receptors. These are G protein coupled receptors present at the parasympathetic neuroeffector junctions. Now, I think we have covered the nicotinic receptors. Now we will be talking about the muscarinic receptors. After we do the receptors, we will then talk about the various cholinomimetic drugs. Muscarinic receptors are 5 types. The N were nicotinic were ion channels. This is the G protein coupled receptor present at the neuroeffector junction at all these smooth muscles and glands. That is precisely where the muscarinic receptors are present. They are associated with various biochemical and electrophysiological responses. See, M1, M3 and M5 are of one type and M2 and M4 are of a different type of G protein coupled receptor. Now just see what does M2 and M4 do. M2 and M4 on interaction with the GI will inhibit adenyl cyclase, decrease cyclic AMP, activation of potassium channels, and inhibition of the voltage-gated calcium channel. It will cause hyperpolarization and inhibition of the excitable membrane. Mainly M2 receptors are present in myocardium. So therefore it will cause bradycardia. So these interact with the GI and cause inhibition of the adenyl cyclase. Inhibition of adenyl cyclase, and there is decreased cyclic AMP, activation of potassium channels, inhibition of the voltage-gated calcium channels. Hence, it is a depressing receptor. It causes hyperpolarization and inhibition of the excitable membrane. Now come to M1, M3 and M5. Now, these receptors, I will talk about their location. This is M1 is mainly in the CNS and GIT. M3 is mainly in the blood vessels, smooth muscles, glands. Okay. Again, I will in detail talk about their location. M1, M3 and M5 act by activation of phospholipase C. There is production of phosphoenol pyruvate, a phospholipid along with the diacylglycerol. They also act by activation of phospholipase A2, the mediators for inflammation. This IP3 causes the ligand-gated calcium channel activation, and there is stimulation. So M1, M3 and M5 are stimulatory receptors, while M2 are depressing, cause bradycardia. So what is happening? IP3 is inositol phosphate 3 calcium-mediated phenomena like contraction of smooth muscles caused by IP3, and diacylglycerol activates protein kinase C and modulation of various physiological roles. Just have a look at it. Phosphoenol pyruvate, phospholipase C is activated. You have production of phosphoenol pyruvate and diacylglycerol. From phosphoenol pyruvate, you have production of IP3. IP3 causes calcium-mediated responses, and here protein kinase C is going to be kinetic, attaching to various moieties and cause excitation. Diacylglycerol activates protein kinase C, modulation of various physiological roles. IP3 causes calcium-mediated phenomena like contraction of smooth muscle and secretion. M1, M3 and M5 activation of phospholipase A2 also happens. Release of arachidonic acid and stimulation of adenyl cyclase and cyclic AMP. So basically by all these methods, IP3-DAG pathway, M3, M1 receptors are excited. Now whatever I was telling, I showed you the diagram of N, N, and M receptors some time back. Now this is a chart which shows integrated properties of muscarinic 1, 2 and 3. M1 is neuronal, M2 is cardiac and M3 is glandular. Neuronal means it is present in the CNS along also in the GIT. Ganglia, it is present at all the ganglia, enteric ganglia, autonomic, GIT and CNS. M2 is present in the heart, SA node, AV node, presynaptic terminal. M3 present in the exocrine glands, smooth muscles, vascular endothelium and CNS. Okay. So, how are they acting? I just told you. Increase phospholipase C, increase IP3-DAG, calcium entry is the main mechanism. Here there is the depression of adenyl cyclase, decreased cyclic AMP, potassium channel opening. Like M1, M3 also increase phospholipase C, IP3-DAG, calcium entry. These are both the three, all the types of muscarinic receptors are G protein coupled receptors. Selective antagonists at M1 receptors are pyrenzapine and tilenzapine. Antagonists at M2 receptors are tripyramine and galamine. Tripyramine and galamine. At M3 is receptor antagonist, that is decrease the glandular secretions, is danephenecin and tolterodine. As I said, M4 and M5 don't have much actions. They're largely confined to CNS. We don't know much about them. M4 is also a presynaptic receptor. M5 is present in neurons in substantia nigra. This is all that we know about them. M4 is mainly presynaptic and M5 in neurons in substantia nigra. So till now, what we have discussed is what is acetylcholine, how it is produced, what are the receptors on which acetylcholine is acting. Well, we understand that it is a very widespread neurotransmitter. It acts on the two types of receptors, nicotinic and muscarinic. Nicotinic of 2 types, muscarinic of 5 types. Okay, I guess by now you understand how and why the actions that it produces are produced. Okay, with this basic knowledge, we will now move on to what is known as cholinergic drugs. Drugs affecting the synthesis, release and action of cholinergic transmitters. At all levels, the drugs that kind of affect the cholinergic transmitter. Inhibition of is something which will inhibit the entry of choline into the nerve cell. So acetylcholine will not be formed. Then there is inhibition of vesicular storage. Vesamicol will inhibit. Botulinum toxin will inhibit the release of acetylcholine at the synaptic clefts. So, promotion of release, just the opposite of botulinum toxin is latrotoxins from black widow spider. See, first we read about hemicolinium preventing the entry of choline, vesamicol preventing the accumulation of acetylcholine in the vesicle, botulinum toxin inhibiting the release of acetylcholine, larotoxins increase the release of acetylcholine. Acetylcholinesterases are, acetylcholine is broken up by acetylcholine esterases. But some things which will inhibit acetylcholine esterases or cholinesterases will be anticholinesterases. So they will be cholinomimetic. They will increase acetylcholine at the synaptic cleft. Then there could be receptor agonists, receptor antagonists, various agonists and antagonists which I just showed you. Okay. So these are the ways by which the cholinergic transmission can be upgraded or inhibited at various levels, at the level of synthesis of neurotransmitter, at the level of storage, release, at the level of their breaking down, then receptor agonists, receptor antagonists. I hope you understand. We'll move on to the next slide. See, acetylcholine synthesis, it is being taken up by the vesicle. From there it will be released. Botulinum toxin inhibits the release. Hemicolinium will inhibit the uptake of choline into the neuron and decrease the synthesis of acetylcholine. Acetylcholine, once it is released, is degraded by acetylcholine esterases. You have anti-acetylcholine esterases like neostigmine, will increase acetylcholine levels. Then you have acetylcholine which is released. Non-depolarizing blocking agents like D-tubofuranin will block acetylcholine at the NM junctions. Depolarizing blocking agents like dexamethasone at the nicotinic acetylcholine receptors. So these are the various ways by which various drugs can either decrease or increase the cholinergic transmission. This previous slide